Okay, thank you, and thank you for the invitation to speak. And I'm going to have to take you far back in time now with this presentation. Before I do so, I'll acknowledge contributions of my co-workers, Richard, Zeebe. I'm going to show you a lot of modeling results today, and Richard's done all of that modeling work. It's a collaborative project with myself and the other people that you see here. It's something we've been working on for many years now, and so the basic idea is we're trying to learn something about the carbon cycle and how carbon emissions in the past may have affected ocean pH, and what we might learn about how it will affect ocean pH in the future. As Thorsten mentioned, there's been a rise in CO2, and this rise in CO2, at least till present, has been mitigated in part by the fact that the ocean is absorbing a large percentage of the CO2, and that's been good for the rise in CO2, not so good for ocean chemistry. As a consequence, the pH has been dropping, and along with that, the concentration of carbonate ion. Of course, that affects the saturation state for calcite in seawater. And this just shows a projection out to 2100 and the associated changes in pH and carbonate ion concentration. So we can see this drop in undersaturation, or a decrease in saturation of seawater. That's 100 years out into the future. You can project this out 1,000 or 100,000 years in the future, and these are the sort of time scales that geologists like to think about. And here's a simulation that was run using a box model. I'll say more about the box model a little bit later, LOSCAR. It's a standard carbon cycle box model set up to study effects of emissions of large masses of carbon, in this case 5,000 gigatons of anthropogenic CO2 being released over about 5 centuries. And of course you get these associated changes in PCO2, surface ocean pH drops. This shows a change in the saturation state of the ocean. The pH is decreasing to less than 7.5. And you can estimate what the effects will be on climate depending on the sensitivity that you assume. With this sort of modeling, we obviously have to make certain assumptions. For instance, we keep ocean circulation fixed, the turnover time of the ocean, vertical mixing more or less stays constant. That's unlikely to happen in the future. As you warm the planet, ocean mixing will probably change. The question is how, and how that will affect uptake of CO2 by the ocean. The biological pump might also be affected with global warming. And then there are things like rock weathering. These are all feedbacks, important biogeochemical feedbacks, that are almost certainly going to impact the ability of the ocean to take up CO2 and ultimately how that affects ocean pH. So there are still quite a few questions or uncertainties about what the impacts will be of emissions. I've listed some questions here, of course. What kinds of changes in pH can we expect? What impacts will these changes in pH have on marine ecosystems? Some of the key variables, as I mentioned, have to do with feedbacks. As I'm going to show you, that obviously the mass and the rate of release of carbon becomes really important in terms of answering some of these questions. And in this regard, ocean mixing becomes really important. Ultimately, it'd be nice to be able to test our theoretical understanding of the carbon cycle with case studies in the past. And we do have case studies. We have one big one. This is the Paleocene thermal maximum. And in fact, the project that I have with Richard and my other colleagues was essentially that, as a project that we proposed to NSF, which was to use this event in the past as a case study of natural sequestration of carbon. So going to take you back in time, 55 million years, to the Paleocene-Eocene boundary. And what you're looking at here is a global stack of benthic foram oxygen isotope and carbon isotope data. And this in some ways represents a general climate trend through the last 65 million years, from the warmth of the early Eocene to the colder conditions of the late Cenozoic. And what you can see here is this trend toward lighter oxygen isotope values or warmer temperatures from the Paleocene into the early Eocene. And superposed on that transition, that long-term transition, is this very short-term excursion. And that turns out to be the Paleocene-Eocene thermal maximum. Associated with that, this is the ratio of C13 to C12 in the ocean. It's one way to think of this curve. Associated with that is this very sharp negative excursion. And I'm going to show you a close-up of this. This is it's actually quite an old record. At the time we thought this was high resolution, but it's now a very low resolution record. But here's that trend in the oxygen isotopes, and that's the warming associated with the PETM. And these are benthic forams from the Pacific Atlantic. And so it's clearly a global signal. We see warming of about 5 degrees centigrade. Associated with that is this negative carbon isotope excursion, about 3 per mil. And this is a very distinct characteristic of the PETM. And what it clearly indicates is that a large mass of C12 enriched carbon was released to the ocean atmosphere system during this time. Estimates of that mass of carbon, just based on the carbon isotopes, suggest that something on the order of thousands of gigatons of carbon. And what confirms that hypothesis is what happens to the ocean carbonate chemistry. This is simply the percentage of calcium carbonate in sediments on the seafloor in one location in the Atlantic Ocean. But what happens during this excursion in the isotopes is that all the carbonate dissolves from the seafloor. And this is very consistent with the ocean very quickly absorbing a large mass of carbon, the pH dropping, the ocean becoming acidified. All right. So we see these carbonate horizons in various locations around the ocean. And it turns out that one of the primary constraints on the scale of ocean acidification, if you wanted to gauge to what extent the ocean became acidified and how much carbon it had absorbed, you'd want to reconstruct the changes in the ocean's carbonate compensation depth, or what we call the CCD. And if you can do this, then you should be able to put a constraint on the mass of carbon that the ocean absorbed. And the best way to do this is to drill depth transects of cores spanning, say, a depth range of 2 or 3 kilometers, representing the seafloor 55 million years ago. And we've done this in various locations, Shatsky Rise in the Pacific and the Walvis Ridge in the South Atlantic. And I'm just going to show you records from the Walvis Ridge. They're essentially the best cores we have, are depth transect. And typically, or I shouldn't say typically, but in the South Atlantic, when you drill down through this layer, 55 million year old layer, what you see is this clay layer with a sharp basal contact and a gradational upper contact. And that's this clay layer just set within the normal sediments that you see on the floor in the South Atlantic till the present day. So clearly something changed. The ocean, the CCD shoaled at this location. Turns out we have other cores. This is the deepest core that we drilled, site 1262, and then we drilled these cores up to 2 kilometers shallower. And this is just the cores arranged by depth from the deepest core to the shallowest core. And this is sort of now a famous figure. And it shows this clay layer here. The blue represents the percent calcium carbonate. So this is 0% calcium carbonate in here. And you can see the thickness of this clay layer shoaling are decreasing with depth. And this just indicates that the CCD shoaled over 2 kilometers, and then gradually recovered, okay? So with this kind of information, we can start to begin to reconstruct the position of the CCD as a function of time. And that's all this represents, showing how the CCD shoaled to shallower than 1,500 meters, and then slowly recovered, taking some 40 or 50,000 years to eventually return to the deepest site. And so this is a very simple measure. It's simply the calcium carbonate content of the sediments. There are other proxies we can look at that tell us something about the saturation state or the carbonate ion concentration of seawater. One of these measures is a very simple one. It's simply the concentration of planktonic foram shells in deep sea sediments. Planktonic foram shells tend to be fairly sensitive to the carbonate ion concentration of seawater, as shown here. You have this nice relationship between the concentration of forams, the greater than 63 micron fraction or the sand fraction, and carbonate ion concentration of bottom waters. And so this is just telling us that we're seeing increasing dissolution. These are from modern sediments. And so what I've plotted next to that calcium carbonate record is simply the percent sand fraction from the same cores. And what you can see is that prior to the dissolution horizon, we have high concentrations at the shallower sites, low concentrations at the deep sites. And this just gives us some constraint on a very important boundary we call the lysocline, a point where you start to get increasing dissolution. So it tells us something about the carbonate ion concentration. And then we have this intense dissolution layers. You'll have to get a new battery for this eventually. And then this recovery interval in here. So this is just another proxy of the saturation state of the ocean. There are others as well that are being developed, boron-calcium ratios, and that are being applied. Now, so we have these observational constraints, and we have them for various regions of the oceans. The important things, we have depth transects in the Pacific, the Atlantic. We have sites in the southern oceans, the Indian Ocean as well. And we can start to use these constraints to start to reconstruct the position of the CCD. This is pre-PETM, and this is during the period of maximum dissolution. And this just shows how the CCD shoals. It's relatively a shallow shift in the Pacific and the Indian. The Atlantic Ocean seems to show a fairly large change in the CCD depth. So with these observational constraints, we can start to try to simulate and reproduce what we have with this observational record. And I don't have time to go into the details of the model. I'll just say it's a box model based on, are very similar to previous versions of these ocean carbon cycle models. It does have carbon isotopes in it. One of the things we can do is actually estimate, if we can get the mass of carbon that's added to the ocean, we can estimate the isotopic composition of that using carbon isotope data, as I'll show you in a second. And this is important because it may actually allow us to identify the source of the carbon that was released. This paper is now in press in Nature Geosciences, so you can quickly follow this up and get a copy and read on the details of the model. And what I'm showing you is simply a simulation, one of the simulations that we've done. And this is sort of a best fit with observational data. And so the simulation involves the input of 4,500 gigatons of carbon. And I'll tell you that the delta C13 of that carbon in this simulation has to be minus 50 per mil. This shows how the carbon is input. And we release about 3,000 gigatons very quickly in a couple thousand years, and then continue to leak carbon for another 40,000 years or so, another 14 or 1,500 gigatons. And when we do this, this is the response of the ocean delta C13. And this is the observational data that we have from cores in the Atlantic and the Pacific. And so that's sort of the best fit with the carbon isotope data. This is the model produced response to the CCD in the Atlantic Ocean as a function of depth, this being the shallowest site and this being the deepest site. And then these are the observational constraints that we have from that South Atlantic site that I showed you. And then this just shows how the CCD is responding in the model. And again, we get this more extreme shoaling in the Atlantic. What we're doing is basically bringing the carbon into the ocean through the Atlantic, all right? And also reversing, and to do that effectively, well, it's recreating a circulation change in a sense. So this is what we've done with this simulation, and it's sort of a best fit with observations that we have. Okay, so I'll just check in my time here. How am I doing? I've got 10 minutes, okay. Max, okay. The constraints on the source of carbon, I just said, from the mass, we want to know the mass of carbon. We have this carbon isotope excursion, and we have the changes in the CCD. And the CCD is sort of the way we get at the actual mass. You can't get a unique solution if you just have just the carbon isotope data. Once you have this, you can take the carbon isotope excursion and estimate the delta C13 of the carbon, which I said in this simulation is minus 50. The other thing that's important to know, of course, is how fast the carbon is being released. And that's not as simple as it seems. The answer to that question becomes complicated by the fact that we're having massive dissolution of our proxy records or sediment records as the event's unfolding. And what these simulations show is the effect of the release time on the magnitude of the carbon isotope excursion, as well as the changes in the CCD, the shoaling, the extent of shoaling in the CCD, as well as the rise in CO2. And so you see in this range of experiments running just from 1,000 years to say 80,000 years or so, the differences that you get in these profiles in this particular simulation. So this is really critical, trying to get some sense of whether it's 1,000 years or 80,000 years. Certainly we can tell the difference between 1,000 and 80,000 years, or at least you would think. I'm going to show you one record from Maud Rise just off of Antarctica. This is what you're looking at are data from a core from, it's just, as you can see, about a meter length of core, which we sampled every centimeter or so. And what you see here are just delta C13 or delta 18O values of individual shells of planktonic forams in red, mixed layer. These are thermocline forams in blue, and these are benthic forams in green. And the nice thing about doing something like this is here's the carbon isotope excursion, all right, in the mixed layer planktonic forams. And we've seen this in other cores, in this location and elsewhere. You get pre-excursion values, so you get to a point where you have a mix of pre-excursion and excursion values. And we never get any transitional values, intermediate values, in the mixed layer planktonic forams. And what that's simply, one way to look at that is this, that the carbon isotope excursion in the mixed layer of the ocean and in the atmosphere was abrupt on geologic timescales. It could have been centuries, 1,000 years, probably no slower than that. Whereas deeper in the water column, in the thermocline or on the seafloor, it looked two things. One, it seems like there's a delay in the isotope excursion propagating to the deep sea, and that it may actually be a little bit more gradual, not as abrupt. And at least for us, this is consistent with the notion that the release of carbon, at least the initial pulse, was fairly rapid on geologic timescales. Now, this site, just like all the pelagic sites, suffers from dissolution. So maybe this is just some sort of artifact. We've looked at terrestrial sections, and often in terrestrial sections, the excursion, at least a good portion of it, tends to be abrupt. The reason why it's important is, again, coming back to a simulation. This is for the modern, for the anthropogenic simulation. That change in surface ocean saturation again, is fairly extreme. It has to do with the rapid rate of carbon release. If you look at the carbon isotopes, you have this effect. In the surface ocean, delta C13, with this very rapid rate of carbon release, goes to this extreme negative before it recovers, whereas the deep sea, the carbon isotope excursion is slower. And this has to do with the rate of release relative to the turnover time of the ocean. In this simulation, it's just 300 years or so, and the mixing time of the ocean is more like 1,000 years. And so that very rapid rate of release causes a reversal in the carbon isotope gradient, and this lag effect between the surface ocean and the deep ocean. So this is sort of an important feature, this reversal in the carbon isotope gradient. It says something about the rate of release, and it's also important a reason for why the surface ocean pH would change so much, because the buffering capacity of the ocean is largely in the deep sea. I'm just going to show you one record from a shelf sequence, New Jersey margin. Here's the carbon isotope record. This would have been relatively shallow, 300 meters or so, and wouldn't have been as affected by dissolution, very high accumulation rates. And again, we see sort of the same pattern in the carbon isotopes, the single shells, and you get these pre-excursion and excursion values, but we don't seem to get any transitional values. And the accumulation rates here are about order of magnitude higher than in the deep sea section. And so this, again, gives us sort of the same sense of this very rapid carbon isotope excursion. And when you start to look at the observational constraints that we do have for the magnitude of the carbon isotope excursion, as well as the shoaling of the CCD in the Atlantic, basically the observational constraints point toward a fairly rapid release of the carbon. You could argue that it would probably certainly be less than 20,000 years, but maybe more on the order of 10,000 years or less. And that's just based on these constraints. So again, and the important thing there is, another important aspect of this, of course, is the change in PCO2. So this is it. This is the simulation where we think we have the best fit, 4,500 gigatons, delta C13 of minus 50. The PCO2 change that we get with this is only 800 ppms, which is a problem because the global warming that we see is on the order of about 6 degrees centigrade. And we would need, to account for this sort of warming, we would need something more like a 2,000 ppm rise in CO2, which means either we've underestimated the amount of carbon that was released, or that the sensitivity of the climate system is more enhanced than we are estimating for reasons that are unclear. So, as far as the PETM, an analog for the future, what we could say is that mass of carbon that was released was at least equivalent to the mass that will be released, if not greater. The rate of release is slower, no question, 3 to 5 times slower, and that the recovery was greater than 100,000 years in terms of ocean carbonate chemistry coming back to normal. We use the same model actually to publish this paper, this perspective, and this is simply just looking at various emission scenarios and the effect on surface ocean saturation. And the point here is simple, that the rate does matter, and the best scenario is one in which you have 1,000 gigatons of carbon being released over 500 years. And in this scenario, you get a pH change or a change in surface saturation that's fairly mild, although the question of mild really depends on which organisms you're looking at. And I think that's obviously an open question. But this was just our attempt to lay out what various emission scenarios might do to surface ocean pH. And again, the model that we use is effectively the same one we use for simulating the PETM. I'm going to stop there because I think we're getting short on time. What I will say is, well, what about the impact on marine calcifiers during the PETM? Well, the only extinction that we see is in benthic foraminifera living on the seafloor, and it's very likely that the changes in pH contributed to their demise. We don't know much about the coral reefs. The record's not that good. It hasn't been studied in that much detail. But what's clear is that there's a large decline in the diversity of reefs and the extent of reefs from the Paleocene into the Eocene. Some data indicate that that may have happened right at the PETM. And then as far as phytoplankton, the calcareous algae, there are huge changes in the assemblages at any given location. What happens is that species that are displaced eventually come back. And we don't know how much of that is related to changes in pH or ocean saturation and temperature, but clearly, again, at any one location, there are these massive changes. The point is a simple one, which is, the changes in pH that occurred 55 million years ago in the surface ocean were probably relatively small compared to what we'll see in the future. And I think I'll stop there. Thanks.
Thank you, Jim. Are there any questions? Fortuna?
Did you consider both calcite and aragonite in your modeling approach, and would that make any difference? Or high magnesium calcite? Or
We didn't.
Try the mic.
We didn't explicitly look at the aragonite saturation horizon, but you can probably get a sense of how that would change with respect to the calcite saturation. So my guess is that with aragonite saturation, you're probably seeing large areas of the ocean that are very close to undersaturation. But no, it's not something that we've looked at just yet. So.
Another question?
Do you have any evidence for other kinds of organisms like zooplankton, pteropods, or anything like that in the PETM?
Yeah, I mean, you've got things like ostracods and, well, the planktonic forams, of course. And with planktonic forams, we don't see any mass extinctions. Just the assemblage or the abundance changes that I mentioned earlier. So, and typically what happens is consistent with either the forams moving deeper into the water column or toward higher latitudes, which to me suggests they're responding more to temperature rather than changes in saturation state. You know, what's clear is that it's hard to get the surface ocean undersaturated, so it's still probably very highly oversaturated for the most part. But no, we don't see any evidence of extinctions in planktonic forams.